at the same time. There we go. Ribosomes, right? We kind of covered all of that. So that was kind of sections 1 through 3 of chapter 4. And then section 4 is starting with the endomembrane system, which we had basically kind of just like walked right up to, right? So we've got the nucleolus and the nucleus, which is in its own membrane. And then there's this other membrane that wraps around the nucleus and then connects to these other membranes. Okay, I was thinking about turning on. Which are called the endomembrane system, right? Because your cell's wrapped in a membrane, and then we've got membranes inside that. Endo's just inside. So the systems of membranes inside your cells are the endomembrane system, right? And they basically are making all the things your cells need. All right, so we're going to talk about all the different structures in the endomembrane system, talk about what they do, and then kind of talk about kind of in as cohesive a fashion as we are able, walk through the entire pathway, right? From the nucleus out through all of these structures. So, come on, why aren't you? There we go. So, like I said, the endomembrane system is just a series of internal membranes, right? Connected to the outer membrane of the nucleus that then kind of come out to the side and start folding like crazy. And so as it's folding, it makes these compartments. And those compartments are the organelles in your cell. It's most of the organelles in your cell, right? And so kind of this, we're going to walk through this system of folded membranes, for lack of a better descriptor, right? So for starters, right, if we kind of picture your nucleus is kind of sitting in this little bowl here. It's got its membrane. It's got the membrane that wraps around it, and it connects then to this giant mess out here. And this mess is your endoplasmic reticulum. It is the largest internal membrane of all of those that we're going to be talking about. And so, like any membrane, right, it's a phospholipid bilayer, right? Those little tail and head guys sticking together, making this membrane. It's all folded up, and it's embedded with proteins that are doing different things, right? And so it's a complex system of tubes. It's a complex network of folds and tubes. And so you get these spaces, right? These kind of internal spaces in the folds of this membrane. And so we call that the cisternal space or the lumen, right? Lumen is just kind of a generic term for space inside of something, right? You've got a gut lumen. It's just the space in your gut. There's a lumen. There's a space inside your endoplasmic reticulum, right? And the lumen, the cisternal space formed inside the ER, I'll bounce back and forth between saying ER and endoplasmic reticulum, depending on how many syllables I feel capable of, right? But this creates the largest compartment of your cell next to the cytosol, right? Like next to the whole freaking cell itself, right? So we've got an ER with the cisternal space, and we divide the ER into kind of 2 chunks. So the part closest to the nucleus, right, is called the rough ER, the rough endoplasmic reticulum, because if you're doing things like transmitting electron microscopy or some such, it looks rough. It's got this weird kind of studded, bumpy appearance because it's covered in ribosomes. All right. And review from Monday, what do ribosomes do? What's their job? They convert, yeah, they translate RNA into proteins. So the job then kind of at large of the rough ER is protein synthesis, right? Which kind of makes sense, right? You've got the nucleolus making ribosomes. They can come out here, tack into this membrane. You've got all this DNA that's coding for mRNA, and that can just pop right out here and start getting synthesized into proteins, right? So the rough ER synthesizes proteins. Outside of that, you've got the smooth ER, a little less foldy, a little more tubey, for whatever it's worth, with relatively few ribosomes. So it doesn't really have ribosomes stuck in the membrane. It's got lots of enzymes, which are proteins, which were likely produced right here and then tacked into the membrane in the smooth ER, right? And so these enzymes, right, enzymes facilitate chemical reactions. Chemical reactions in particular are synthesizing, we're making carbohydrates and lipids, things like steroids, right? So we're making all the proteins in our rough ER. We're making our carbs and lipids in the smooth ER, right? Our nucleic acids are in the nucleus. So here's kind of all of our 4 biological macromolecules all sorted out to their respective spaces, right? And since we're making lipids and things out here, this is where we assemble membrane lipids, right? Because you occasionally need to repair or replace things in a cell, right? So your smooth ER is building membranes. It's putting proteins in the membrane that it needs to do stuff. Also, smooth ER side hustle, it also detoxifies substances. So it's making carbs and lipids. It's building these membranes out of proteins from over here and stuff from over here. It's also removing toxins that are coming into your cells, coming into your body, right? And so this means that some cells, depending on where they are, will have a lot of smooth ER. Your liver cells has a lot of smooth ER, right? Because your liver is taking out any bad stuff that you might be consuming that needs to be gotten rid of, right? So smooth ER also works in detoxification. All right. Next, right? Nucleus, rough ER, smooth ER, one last patch of folded stuff. This is the Golgi apparatus or Golgi body, whatever you prefer. So the Golgi apparatus, again, contains cisternae, similar to the cisternal space you see back here, right? And it's just sacks of membrane in the Golgi apparatus, right? It's all just folded up membrane, so it's got little spaces inside of it. And so if the nucleus is like kind of our headquarters with all of the information going out, the endoplasmic reticulum is kind of like your factory, right? It's producing all the stuff. Golgi apparatus then kind of becomes your distribution center, right? So things that are made in the rough and smooth ER are sent in little vesicles, little kind of membrane-bound packets to the Golgi apparatus. The Golgi apparatus collects those things, packages them up, and distributes them either around the cell or to other cells. Right. And so this happens kind of in orderly fashion, right? So things are being made in the rough and smooth ER. They're packed into these little transport vesicles. So basically little pieces of the membrane, say in the ER, kind of enclose whatever is being made, pops off and travels, right, in this little packet to the face of the Golgi apparatus that is facing the endoplasmic reticulum, right? And that's called the cis face. So the cis face of the Golgi apparatus is close to the endoplasmic reticulum. These little vesicles enter at the cis face. They go into the cisternae, the Golgi apparatus, right? And they get modified. If there's any last little finishing stuff that needs to happen, it happens in the Golgi apparatus. Maybe little glycoproteins or glycolipids, little sugars get tacked onto stuff, right? That can happen in the Golgi apparatus. And then opposite of the cis face is the trans face. It's closer to kind of the outside of the cell. And so again, you've got vesicles, right? Little pieces of membrane that close around and kind of pack up the finished thing. And they leave, and either they move to where they need to go in the cell, or they go over to the edge of the cell and are released out into the body. Just depends on what it is and where it needs to go. We've got Golgi apparatus with a cis face kind of towards the center of the cell and a trans face towards the outside of the cell.
So the trans face, you said it pretty much is on the outside of the cell, right?
Yeah, it's like facing out. So the cis face is kind of back towards the nucleus in the ER, and then the trans face is facing like out towards the cell membrane, or kind of away from the nucleus. Right, so this is kind of like another image of the same thing, right? Just kind of like angled a little differently, right? So we've got the nucleus, you've got the rough and smooth ER that's kind of wrapped around it. Things are passed through, proteins, lipids, carbs, all that stuff are released in these little packets, these little transport vesicles, right? You've got the cis face, which is the part that's facing back towards the nucleus. These vesicles enter in, stuff gets finished, gets kind of packed up in these little secretory, like secretion vesicles, right? So they can be released and used wherever it is they are needed. All right, so that is kind of, in broad strokes, the protein synthesis pathway through the endomembrane system. All right. So another thing that the Golgi apparatus does is it makes lysosomes. So it's just another little vesicle, another little kind of membrane-bound packet, but these carry enzymes specifically, right? So they're digestive vesicles. To lyse, like if you lyse a cell, you break it down. So lysosomes are little vesicles that carry digestive enzymes, right, to break down things. So they fuse with things like food vesicles or old organelles that need to be repaired or replaced, right? And those degrading digestive enzymes inside of them can break down things like old organelles, right, to their kind of component macromolecules, whatever they were made of. And those can be recycled. They can be used to make other things in the cell at that point, right? They're really important, kind of as with anything, even these little kind of tiny parts of this system, if something goes wrong, can have kind of large downstream physiological effects. So you can have lysosomal storage disorders, right? Because lysosomes are kind of cleaning up, getting rid of old stuff, getting rid of stuff that needs to be gotten rid of. So things like Tay-Sachs disease, you've heard of Tay-Sachs. Tay-Sachs is a lysosomal storage disorder. So it's just losing one of these lysosomal enzymes that's used to break stuff down. Specifically, the enzyme that would break down glycolipids that are building up in your nerve cells, right? And when those glycolipids start to build up, those nerve cells lose function, which causes those symptoms in Tay-Sachs, things like seizures and muscle rigidity, right? So it's due to kind of a dysfunction in the lysosomes specifically. All right, so we've covered the endomembrane system. We've talked about that being your organelles, but it's actually not all of the organelles in your cells. There are these two guys that are kind of different. They're separate from the endomembrane system, right? And those are mitochondria and chloroplasts. Chloroplasts obviously being specific to plants, but they kind of fall in the same category. So we're going to talk about the structure and the functions of mitochondria and chloroplasts, kind of talk about how they're different, how they're similar. They are very similar, and how they're different, right? And then talk about where they came from and why they're not part of the rest of your kind of system of organelles in your cells, right? So both mitochondria and chloroplasts are surrounded by a double membrane, right? So kind of like the nucleus is, there's one membrane and then they've got a second membrane around that. They also contain their own DNA. Because they contain their own DNA separate from your nucleus, they can make their own proteins, right? So they kind of function independently. Their function, as far as being part of our cells, is energy metabolism, right? If you covered this in high school anywhere else, right? We always call mitochondria like the powerhouse of the cell, right? They can break down glucose for energy, right? But they're not autonomous. So even though they're kind of double bound in their own membrane, they've got their own DNA, they do their own division during mitosis, right? They kind of handle themselves. They are completely dependent on our cells. They can't function. You can't take a mitochondrion out of a cell and have it exist on its own, right? It's lost that ability, right? So they're both involved in energy metabolism, surrounded by a double membrane with their own DNA. But they're not exactly the same. So mitochondria, right? We have everything has we and plants both have mitochondria. But mitochondria metabolize sugar to make ATP, right? And ATP is kind of what powers a lot of cellular functions, right? So like I mentioned, it's bound by two membranes. There's an outer membrane, just kind of this oblong, smooth membrane that kind of is holding the whole thing. Inside is this inner folded membrane, okay? So you've got an outer membrane and then an inner membrane that is folded, and these folds are called cristae, right? They kind of make these separate little sections of the inner membrane. And so in here, inside this folded membrane is called the matrix, right? And then you end up with these spaces between the inner membrane and the outer membrane. And it's called the intermembrane space, right? Inter means between. So it's the space between the membranes is the intermembrane space. Inside you have all this other stuff that's called the matrix inside the inner membrane. And then we've just kind of got this outer capsule-like membrane. All right, so that's mitochondria. Fairly simple, really. And then, of course, their own DNA. Their DNA is circular. Interested? And so relatively simple structure-wise, as opposed to chloroplasts, which actually have a relatively complicated structure, a more complicated structure than mitochondria, at least. And so chloroplasts, right, we find these in organisms that photosynthesize, right? They take light energy, they generate ATP and then sugars, right? So they can take light and eventually convert that into ATP and glucose. So they're larger than mitochondria and they're more complicated structurally, all right? So we still have, right, the smooth outer membrane and a smooth inner membrane. So we've just got these two membrane layers, right? It's just kind of double wrapped. But then inside of that, there's a whole other membrane system, kind of its own endomembrane system. So we've got these little things, they look like little coins or pancakes or something. So these little closed kind of compartments of membranes, like I've just taken a membrane and made a little flat package with it. So those are grana. And these grana are stacked, like a little stack of pancakes, right? We call those stacks thylakoids, right? So you've got little membrane packets, grana, stacks, thylakoids, right? And so you've got all of these thylakoid membranes, right? Stacked all over the place. And of course, we've got some fluid matrix around that. That's called the stroma, right? And so when we get to photosynthesis, we'll kind of talk about this a bit more, but these membranes, right, are what photons are hitting when we're converting light to ATP. And then that energy is going out into the stroma, and the stroma is where the enzymes for synthesizing glucose are. Right, so like I said, we'll unpack all of that in detail in the photosynthesis chapter, but this is where it's happening. So chloroplasts, similarly, they are energy generating like a mitochondrion, but they are more complicated. So where'd they come from? So the theory that explains where mitochondria and chloroplasts come from is called Endosymbiotic Theory. It was kind of proposed and highly researched, well, by several people, but one of the kind of very strong people back in, I think, like the 70s was Dr. Lynn Margulis. Interesting lady, thought some weird things, but she was square on this. She thought some weird stuff, but she was fine with the endosymbiotic stuff. She was good at this. Interesting lady. But basically, endosymbiotic theory states that we have our eukaryotic cells with mitochondria and chloroplasts, where appropriate, due to one living cell being engulfed and becoming part of kind of our ancestor eukaryotic cell. All right. So basically, some little single-celled kind of eukaryotic ancestor of all of us was bopping around, and if you ever look at single-celled eukaryotes, right, they eat by kind of taking their membrane and wrapping things and pulling them into the cell. It's called phagocytosis. We'll talk about it in chapter 5, right? But they engulf these things, right, into these kind of, they make them into their own little vesicle, and then they digest them, right? They break them down and consume them. But basically, this theory states that what happened was there is an aerobic prokaryote, right, something capable of oxidative metabolism, that was engulfed by one of these eukaryotes that did not have mitochondria. And instead of getting digested, it just kind of hung around, right? So it was engulfed, it was wrapped in a second membrane layer, which is why mitochondria have two membranes wrapped around them. But then it just bopped around. It never got digested, it never got away, right? And the eukaryotic cell began using the products of this little cell's metabolism for its own purposes. And so there was kind of a symbiosis formed. And eventually, over a long period of time, right, this cell eventually was no longer kind of its own entity. It couldn't get away anymore, right? So now they're just kind of a component of eukaryotic cells. And the same thing with the chloroplast, right? A eukaryotic ancestor of all the plants, already in possession of a mitochondria at this point, engulfs some photosynthetic prokaryote, right? Trapped it in a little layer of membrane, giving it its kind of double membrane. It didn't get digested, formed a kind of a symbiotic relationship with the eukaryote, and eventually became kind of an integral component of that cell, of that organism, right? And so this is why, right, we talk about kind of the endomembrane system. Everything's kind of integrated and all wrapped in this internal membrane and together, except the mitochondria and chloroplast, because we got those in our cells differently, right? They weren't just kind of integral to the cell from its beginning, right? So origin of mitochondria and chloroplast through endosymbiosis. All right. So that's all of our interior membranes, right? So we started in the nucleus. We've walked through all these internal membranes and organelles. All right. And so around all of those membranes and organelles, right, we need something to help us move things around and hold things in place. And that is the cytoskeleton, right? Cyto just means cell. So it's the cell's skeleton. That's what we're talking about, right? So we're going to talk about a few different cytoskeleton fibers, kind of what they look like, what they do, and talk about, I mean, they do, for one, provide structure, but they also are able to move things around the cell. So we'll talk a bit about that also. So cytoskeleton gives the cell its shape, right? Makes the cell roundish or like a red blood cell. The cytoskeleton is what makes red blood cells, that kind of weird little donutty shape. It will anchor, it'll hold organelles, right? Pieces of the endomembrane system or a mitochondria where it needs to be in that cell. But they're not, generally speaking, kind of static or permanent structures. Your cells are actually kind of building and breaking down cytoskeleton pieces all the time, depending on where things need to be or what they need to be doing. So your cytoskeleton is kind of constantly assembling and disassembling. All right, so we're going to talk about 3 different kinds of fibers, right? Actin filaments, microtubules, and then intermediate filaments. So actin filaments, sometimes called microfilaments, they're the tiniest fibers of the things in your cytoskeleton. And so they're basically just like tiny little threads. They're just made of 2 chains of actin subunits, right? So actin's a protein. You make a long chain of little actin proteins. You kind of coil it up with a second chain of actin proteins. And so you end up with this kind of thread-like filament, all right? Really thin, but they're used a lot. They're involved in cell movements, which we'll talk about in the next section, and cellular extensions, right? So ways that the cell kind of changes its shape and moves around in some cases. So we've got actin filaments, smallest filaments, just 2 little kind of chains of actin twisted together, right, to make one of these fibers. The largest fiber is the microtubule, right? And these are kind of think of them like pipes or like tubes, like small tubes, hey, right? And so don't get too bogged down. So they're made of tubulin, right? Actin filaments are made of the protein actin. Microtubules are made of the protein tubulin. But basically the tubulin forms these rings, and you put a lot of these rings together and you get this kind of pipe or tube-like structure, right? And so these actually, even though they're the largest, are kind of the most in flux. They're constantly being built and taken apart and built and taken apart, right? They're frequently used kind of in my head, kind of like little railroad tracks, like they're built and things are moved along them and then we just kind of take them back down again, right? So used in cellular movement, organizing cytoplasm, kind of pushing things where they need to be, and then moving materials, products, proteins, and things around the cell. All right. Lastly, intermediate filaments, right? We've got our smallest filament, our largest filament, and then the intermediate filament, which is kind of in between the 2. Don't get too bogged down in this. Just know that it's got a pretty complex structure, right? It's proteins wrapped around proteins, interlocked with proteins, made into a big sheet, rolled up into a big thing. Basically, it's just a really durable element of your cytoskeleton. It's kind of the takeaway from all of that, right? And so it's complex. It's like this giant kind of filled in big, thick rope of kind of protein fiber. And so these generally, of the three, are the ones that aren't broken down. So once intermediate filaments are made in the cell, they stay in the cell, right? Which kind of makes sense. It looks like something that's a lot of work to make your cells, not just going to be like popping these apart and putting them back together. Right, so they're kind of medium sized, but they're very, very durable. We see them in things like vimentin or keratin. Vimentin provides kind of structural stability to a cell, so kind of like scaffolding, maybe, if you think of it that way. And then keratin is one we're more familiar with. That's what's in your skin. It's what your fingernails are made of. Your hair are all kind of made of this. Keratin is a main component in a lot of those sorts of cell products, I guess, or tissues. No, not tissues. Right, so it's it's a tough substance. All right, so we've got these three cytoskeletal fibers. All right, some kind of extra ways in which they're used that will come up later. So this is kind of looking forward to chapter 10. So microtubules become big in mitosis. So in your cells, when they're getting ready to divide, they've got these kind of regions at each pole called the centrosomes. And so these are kind of where microtubules that are going to be separating your chromosomes start to get organized, right? So you've got these big microtubule structures. These are centrioles, right, that are kind of at each pole that attach to chromosomes that will be pulling them apart, right? And so microtubules in particular are really important in cell division. We've got things like, again, microtubules moving things around. So they're pulling chromosomes to the opposite sides of the cell. In the case of the centrosomes, you've got actin filaments, right? Those are what are sliding in your muscle cells, right? That cause muscles to move. You've got motor proteins, right? And they're kind of what are riding along on these little microtubule railways as they're built so that products from your endomembrane system get where they need to be, right? They're moving vesicles around once they're produced either at the smooth ER or the Golgi apparatus, right? So they're holding things in place, they're transporting things around, they're moving things when cells are moving or dividing, right? And so let's talk a bit about movement. So we're gonna talk about how cells move. There's a couple, 3 ways we'll talk about as far as how cells move. And then we'll talk a bit about the extracellular matrix that is present in animal cells. We'll get there. But first, cell movement. So, first way we see cells moving is crawling. This is kind of like amoebas have this kind of oozing, crawling sort of motion. And so you have a cell that for whatever reason needs to move. So it produces these actin filaments that kind of push the plasma membrane forward. And as the membrane is moved forward, they fill that in with microtubules to kind of give it a bit more structure, right? And then these myosin motors with more protein kind of motors are able to pull the cell towards the part they just stuck out. So they can kind of ooze things out with filaments, fill it with tubules, and then pull themselves forward. So you get this kind of oozing motion. We see this in things like white blood cells. So white blood cells get released, they're blood cells, released into your bloodstream, right into your circulatory system. They crawl out of your venules, kind of the ends, the tiniest ends of your veins, right? And then they've got to find whatever foreign body or cell or pathogen they're after, right? And so they crawl in this way and eventually get to whatever icky thing is in your body that they need to get rid of. Yep.
Is that what pus is?
Pus is in part spent white blood cells, I believe. There's other stuff in there. But yeah. But part of, I believe, what is in pus is white blood cells. Yeah, it's gross. That's fair. All right. So they can crawl. They can swim, right? I mean, we've got flagellated cells that aren't swimming, but there are single-celled eukaryotes that rely on things like flagella, which are long structures made of microtubules, right? Specifically, not that you need to know much about a flagellar structure, but if you took a cross-section of a flagellum, it's actually 9 pairs of microtubules with 2 microtubules in the center, right? And so these tubules kind of sliding around each other is what allows the flagellum to wave. And so something like this euglenid will wave its flagellum kind of from the end and pull itself through whatever medium, usually if they're usually aquatic, will pull it through whatever it's in. All right, so you can have organisms that have this kind of singular long, or sometimes 2 flagella. You can also have organisms with things that are like lots of tiny short flagella. We call those cilia, right? So structurally, like if you took a cross section, they look a lot like a flagellum, but they tend to have lots of them. If you're looking at these, right? This is a skimming electron micrograph. They look kind of like fur, but again, they're full of microtubules, so they can be waved. So they tend to be kind of waved synchronously to allow some organism to move through its environment, or to line your gut to move stuff through your gut, or whatever else you need things to be moved for, right? So things can crawl, cells can have flagella, cells can have cilia, and they're all kind of controlled and structured by these cytoskeletal elements. All right, so we started in the middle, walked through all this membrane mess. We tacked in chloroplasts and mitochondria. We held it all in place with this cytoskeleton stuff. So we're basically at the outside of the cell, functionally. So we'll have a little plant sidebar, right? Because our animal cells, that's kind of where it stops. We've got this membrane, and that's, as far as the cell itself, that's what we've got. In plants and fungi and some protists, you have a cell wall, so something else, right, that's giving the cell even more structure. So in plants and protists, tends to be made of cellulose. We mentioned this when we were talking about biological molecules. In fungi, it's chitin, that kind of interlaced carbohydrate protein situation. But in plants specifically, we'll talk about plants, cell walls specifically, right? You've got this little plant cell, so it divides, it's forming, it's growing. And as it's growing, it creates a primary cell wall made of cellulose, right? So while the cell is forming, so it's kind of this thinner outer layer. Once that plant cell has gotten as big as it's going to get, it kind of fills in, right? Primary means first, it fills in a secondary or second cell wall inside of that. So kind of think like tree rings, right? You've got a primary cell wall. When it's done growing, it fills in that inner secondary cell wall. And then on top of that, plant cells are all about control, right? They stick each other together using this middle lamella. So it's kind of this sticky layer that forms between the primary cell walls of adjacent cells, right? And it glues the cells together. So you have a cell, secondary cell wall, primary cell wall, lamella sticking them all together. But that's in plants. We're not plants. We don't do that. So we've got to have some other things going on. So in animal cells, right, we've got our little Golgi cell membrane. And outside of that, we have what's called the extracellular matrix. So it's a matrix, a bunch of stuff that's outside the cell. All right, so we've got an extracellular matrix. And it's a mixture of glycoproteins, right, proteins with those little kind of sugar tags on them, that's stuck around the outside of an animal cell, and it's connected to the inside of the cell, generally to the cytoskeleton, right? So if we've got this membrane here with all of its little membrane proteins and stuff stuck to it, you've got this extracellular matrix, all these proteins and little sugar chains and things, and they're actually connected through the membrane to the cytoskeleton on the inside. So it's kind of like antennae or sensory organs for the cell, right? So you can have mechanical and chemical signaling between this ECM, the extracellular matrix, and the inside of the cell. So it kind of allows the animal cell to sense what's around it, to react to what's around it. All right, so we've got this extracellular matrix around animal cells. All right, so we've gotten through all of the bits of cells, right? Middle of the cell, stuff in the cell, cell membranes, walls, extracellular matrices. We're going to take matrices. We're going to take it one more step and we're going to connect cells to other cells, right? Because then we get, right now we have a thing. We've connected the cells together. You start making tissues and creatures, right? We're almost there. Oh yeah, we totally got this. All right, so we're going to talk about cell junctions, things that connect cells, and then talk a bit about surface proteins. So surface proteins are like name tags for cells functionally, right? So you've got these proteins attached to the surface of a cell, and it's got some molecule stuck to the end of it that says what that cell is, right? So this allows cells that are the same type or belong to the same tissue or to the same organism at least to recognize each other. They can connect to each other. They can coordinate their metabolic and various synthesizing functions, right, and work together, right? So everybody's got a name tag, says what kind of cell they are. Most of these are glycolipids. So lipids with little carbohydrate heads that kind of signal what kind of cell it is. For example, you've got glycolipids stuck to the outside of your red blood cells. This is what gives you your blood type, right? So if you've got type A blood, you've got these type A glycolipids attached to your red blood cells. And that's why when you get a transfusion, you need to make sure you get type A blood, because if you get blood cells basically with the wrong name tags on, your body gets really angry because they look like they don't belong. They look like they belong to some other organism, right? And then your body starts attacking itself and it's not a good time, right? So surface proteins kind of identify cells as certain types. All right, so if cells are of similar types, for example, in the same tissue, it's ideal that they'll be connected together somehow. We are animals. We don't have middle lamella or other things. So we're going to talk about connections between cells. So 3 kinds of junctions. We'll talk about them fairly briefly. So adhesive junctions, tight or septate junctions, and then communicating junctions. So adhesive junctions. Adhesive junctions are found in all animals and are just what they're said, right? We don't have a middle lamella, but we have adhesive junctions, right? These things that just kind of, the way this picture looks like Velcro, right? Just stick the cells together, right? So they're attached either from cytoskeleton to cytoskeleton, right, of 2 different cells, or from cytoskeleton of one cell to extracellular matrix of another cell, right? Somehow they're using those fibers to attach each other to each other. You frequently find these, or you find more of these in tissues under stress, right? Which makes sense, right? I really, really, really want my muscle cells and my skin cells to really be attached to each other. It would be really unfortunate and pretty gross if they weren't, right? So you've got loads of these adhesive junctions making sure your cells stay attached. So we've got adhesive junctions. We've got tight or septate junctions, right? And so these kinds of junctions create watertight layers of cells. So anytime that you want something to be watertight, you want to make sure that whatever is lined with these cells isn't going to leak, right? Things like your gut, right? You need these tight or septate junctions, right? To make these really, really tight connections. So both vertebrates and invertebrates have what are called septate junctions. In vertebrates, we have another kind of junction that works similarly called a tight junction, right? So cells lining things like your digestive system, where you absolutely need to make sure things don't leak out, is lined in cells that are connected with tight or septate junctions. All right, last one. Communicating junctions do what the name says. They allow communication between cells. Generally by osmosis, right? Cells don't talk, but they can pass chemical signals between each other. So you can have junctions that allow molecules to pass between cells that can allow communication, right? So in animals, we have gap junctions. So they can be open or closed, right? So if a cell needs to tell a cell something, you can open the gap junction, pass a chemical signal through, and then close the junction. In plants, it's a little different. They have what are called plasmodesmata, which is a weird word. but these are membrane lined. They are always open channels. So basically plant cells are kind of like always talking to each other. They basically have these tunnels between them, these plasmodesmata, right, that connect the endoplasmic reticulum of both of the cells. So plant cells are better communicators than animal cells? Maybe, I don't know. They're definitely more open, right? So we've got these communicating junctions, either gap junctions in animals or plasmodesmata in plants. All right. So we've built up our cell. We've connected our cells to other cells. We have mastered cells. And that is the end of chapter 4. All right. And so I'm going to go ahead and leave us there. We will